The Squamata, or squamates, are a clade of the scaled reptiles, or Lepidosauria, containing around 11,000 species. The traditional terms lizards and snakes are used to refer to various squamates, but are misleading because snakes are phylogenetically nested within the lizards. That is to say, snakes are just one successful group of legless lizards. Numerous other groups of lizards have also independently lost their legs. Squamates are amniotes, that is, they have shelled eggs with an amnion. Most species are oviparous, that is, they lay these shelled eggs, but many species have evolved viviparity, that is, they give birth to fully developed young. Unlike anamniotes, squamates do not have a larval stage and do not undergo metamorphosis outside the egg. The order Squamata is divided into several major suborders. The taxonomy of these suborders has been in flux as historical morphological cladistics was overturned by DNA-based phylogenetics. This means that many traditional groupings or hypothetical relationships were wrong. This especially affected limbless squamate species because evolution of limblessness is associated with homoplasy, or convergent evolution, of other traits. This video will cover comparative internal and external anatomy of several different major groups of squamates and techniques relevant to research on them. Our first study object is Phelsuma standingi, Standing's day gecko. This is a gecko species from western Madagascar that is adapted to dry forest habitats. These are some of the largest living Phelsuma species, but our study subject is a subadult. Geckos belong to the infraorder Geckota. Most species are covered in small, fine, granular scales, which are often larger on the belly, though some species have very large scales. Along the lips, most species have larger scales. Geckos are perhaps most well known for the pads on their toes. These pads are divided into rows, called lamellae. Each lamella is covered in microscopically finely divided hairs that allow surface adhesion through electrostatic interactions, or possibly van der Waals forces. Phelsuma do not have claws, but many other gecko species do. The squamate hand and foot are ancestrally pentadactyl, that is, they bear 5 digits. In Phelsuma, the first toe is reduced. Like most tetrapods, but unlike mammals, squamates have a single multi-purpose excretory orifice, the cloaca. Dentition is an important means to identify major groups within Squamata. Geckos have pleurodont teeth, which means the teeth are attached to the inner surface of the jawbones and can be replaced. Most geckos do not have eyelids, but instead have a special scale, sometimes called the brille or ocular scale, that covers the eye. Geckos have external ear openings, in which the thin tympanic membrane is visible. In geckos, pupil shape often correlates with activity period. Species with round pupils are typically diurnal, and those with slit-shaped pupils are typically nocturnal. Phelsuma standingi has round pupils, though you cannot see them in this specimen. To prepare the gecko specimen, we place it in the supine position in the dissecting tray. It is pinned in place with a pin through the tip of the tail and each limb. To dissect the specimen, we make a sagittal incision running from the posterior abdomen to the pectoral region, or chin. We then make lateral incisions at either end of this sagittal line. The skin of the trunk is pinned to either side, taking care to separate the viscera from the skin by running a blunt probe along it as it is pulled away. To open the body cavity, a small incision is made posteriorly and run anteriorly along the sagittal line. We must cut through the thin, semi-translucent xiphisternal and post-xiphisternal ribs. We can then gently pull open the peritoneum to expose the internal organs. The sides of the peritoneal space can then be pinned in place. We further cut through the rib cage anteriorly to access the heart and other anterior organs. We have to gently cut through the viscera to free the heart. We can now examine the organs of the gecko. A blunt probe is used to manipulate the organs without tearing anything. The large reddish-yellow structure is the liver. Dorsal to it runs the stomach, followed by the small intestine and large intestine. The heart is small and anterior. The lungs of this specimen are small and collapsed. The gallbladder can be seen as a small greenish extension on the ventral surface of the liver. We extend the digestive tract somewhat to be able to follow its course. We extend our dissection line posteriorly to the cloaca in order to examine the excretory and reproductive systems. The kidneys are visible as thin strips that lie along the ventral surface of the spine. Because this individual is not sexually mature, the gonads are small. Now we examine the gular region of the gecko. An incision is made along the chin so that the skin can be separated. Through the translucent muscular floor of the chin, we can make out the trachea and esophagus. In many reptiles, calcium sacs are present just behind the quadrate bone. These are used to help support bone development and by females also to produce eggs. The calcium is kept in a fluid solution so that when it is cut into, the bright white or pinkish white fluid escapes. The heart of squamates is divided into 4 chambers. Like the amphibian heart, the division of the left and right ventricle is not complete, so that there is some mixing of oxygenated and deoxygenated blood. Oxygenated blood from the lungs enters the heart from the pulmonary vein via the left atrium. It enters the left ventricle, mixes somewhat with deoxygenated blood, and leaves the heart via the aorta, which is divided to run left and right. After traveling through the body, deoxygenated blood enters the heart again via the right atrium, and is then pumped by the right ventricle to the lungs via the pulmonary artery. To give an idea of some of the variability of geckos, we will briefly present Rhacodactylus auriculatus, the gargoyle gecko. It is a representative of the Diplodactylidae, a different family of geckos from Phelsuma. These geckos are from New Caledonia. The foot of Rhacodactylus auriculatus also has undivided adhesive lamellae, in addition to large, powerful claws. Returning to Phelsuma, we will now look at a specimen of another species, Phelsuma quadriocellata, the peacock day gecko. This species is much smaller than Phelsuma standingi. This is an adult male. Notice the differences in coloration. Males of many squamate species have femoral pores on the thigh. These secrete a waxy substance that emits pheromones and is involved in communication, especially between the sexes. Our next object is the bearded dragon, Pogona viticeps. This is a representative of the family Agamidae, the agamid lizards. Bearded dragons are from Australia, but are very popular in the pet trade. They are omnivorous lizards adapted to arid landscapes. The scales of Pogona viticeps are imbricated, that is, they overlap one another, and they are keeled, so they have a central ridge. Scale shape in squamates can be important for taxonomic purposes. Unlike the geckos, which have pleurodont dentition, agamids are acrodont. That means that their teeth sit on top of the jawbone, not along its lingual margin. Notice also that, unlike the geckos we have seen, the bearded dragon has full eyelids. The spines on the head and flanks of bearded dragons are specialized scales. The procedure for dissecting the bearded dragon is just like the Phelsuma dissection. We begin with a sagittal incision, starting at the hip and running to the breast. Here we have already cut through the abdominal wall. We then pull the skin away from the underlying tissue. We can do this by hand because the specimen is large and robust. Immediately note how much smaller the rib cage of Pogona viticeps is than in our gecko specimen. Notice also the thick pectoral muscles. As before, the skin of the body is pinned aside. Now, with the rib cage fully opened and the peritoneal wall pinned aside, we can see the full organs of the bearded dragon. Note that the peritoneal membrane of the bearded dragon is almost pure black. This is probably an adaptation for thermoregulation. To better examine individual organ systems, we must free them. First, we separate the mesenteric membranes that connect the intestines and other organs using a combination of scissors, tweezers, and a probe. We can then extend the organ systems to the side of the specimen to see the full system. The stomach of the bearded dragon is large and distensible. This specimen has large, bright yellow and brown fat bodies. These stores of energy can help it to survive during periods of relative starvation. The kidneys and testes are both large. They sit atop one another along the spine. We can remove the heart to examine it in more detail. Here we can see the right atrium, left atrium, right ventricle, left ventricle, and aortic arch. We will now prepare a female Cameliocalyptratus, the Yemen chameleon. Our subject is an adult female. Chameleonidae, the chameleons, are the sister group of the Agamidae, and share with them the acrodont dentition. Together they form the clade called acrodontia. Chameleons have numerous unique characteristics, such as their unusual foot configuration with two bundles of toes, their non-autotomizable, generally highly prehensile tail, independently moving eyes, ballistic tongue, and pronounced color changeability. Also, their internal anatomy is quite unusual because the body of most species is laterally flattened, so that it is very narrow but also very deep. For this reason, dissection of chameleons is carried out in lateral, not supine position. We can observe that the bundles of the hands and feet are different. The hands have a bundle of 2 outer and 3 inner digits. The feet, on the other hand, have 3 outer and 2 inner digits. On the head, we can observe a series of bony ridges, as well as a pronounced crest of long, pointed scales along the chin. Note that chameleons do not have an external ear. This is a stark contrast to most other squamates. The top of the head bears a structure called the casque. This is formed by elongations of several skull bones and also has important muscle attachments. To prepare the chameleon from the left side, the left forelimb and hind limb are removed. A shallow incision is made along the left side of the ventral midline, and we work the skin away from the viscera using probes, scissors, and gentle pressure. We can then cut away this flap of skin. Chameleon ribs extend nearly the full length of the body. Chameleons often break their ribs in the wild and in captivity. To access the internal organs, we now cut away the left ribs. This can be done with sturdy scissors. The ribs are divided into 2 segments: the vertebrocostal segment, which is bony and runs postero-ventrally, and the sternocostal segment, which is cartilaginous and runs antero-ventrally. Postero-ventrally, there is a large pinkish fat pad in this specimen, which can be lifted away to reveal the posterior organs. The lung of the chameleon is large and can be used to inflate the body during threat displays. We can lift it forward to see the digestive system beneath. The liver is ventral and positioned to the right side of the body. The ovaries here are full of egg follicles. The gallbladder is slightly greenish. The intestine is dark and tightly bundled. The rectum runs ventral to the ovaries. We now pin the animal in place in order to manipulate the organ system. To access the digestive system, we cut through the mesenteries. The heart lies far forward in the body. The stomach is large and distensible, able to cope with large, mostly unprocessed prey items. The pancreas is visible as a thin organ that runs alongside the stomach. The gallbladder is relatively large and is closely connected to the liver. The kidney sits dorsal to the ovaries. To examine the heart, we extend our incisions slightly forward. We can then excise it to look at it in more detail. We next want to look at the ballistic tongue mechanism of the chameleon. To access it, we remove the skin from the gular region. We need to break through the quadrate bone using wire cutters in order to remove the lower jaw. At the anterior end, we cut through the medial symphysis of the two lower jaws. We can now see the tongue and hyobranchial apparatus that is associated with its action. The tongue is bunched up on the lingual or entoglossal process of the hyobranchial apparatus. Posteriorly, there are two long arms, called the ceratobrachials. By holding the tip of the tongue and pulling the specimen away, we can see the full reach of the tongue. The tip of the tongue is thick and muscular. It has, at its distalmost end, a fleshy and sticky pad, which engulfs prey items and sticks to them, to then draw them back into the mouth. With the lower jaw removed, we can also examine the palate of the chameleon. Here we can see the palatine and vomer bones, which form the roof of the mouth. The anterior slits are the openings of the vomeronasal organ, and the posterior cleft is the fleshy palate. A very important technique in reptile taxonomy is the preparation of the hemipenis. These are the reproductive organs of male squamates, which are always paired. Here, we will demonstrate this technique using an adult male Hoenel's chameleon, Trioceras hoeneli. Trioceras hoeneli is a small chameleon from Central Eastern Africa. Males can be identified by the pronounced hemipenial bulge at the base of the tail. To prepare the hemipenis, the deceased reptile is held in one hand, while manipulation and preparation are carried out with the other. For left-handed people, it may be convenient to mirror the approach shown here in right-hand view. Note also that preparation technique differs a little depending on the squamate group being prepared, because lizards that can drop their tails require a little more care during manipulation. First, locate the cloaca and the extent of the hemipeneal bulges. Pull gently with the holding hand, here the left hand, to expose the cloaca. To make it easier to prepare the hemipenes, it is important to prime them for expression. We do this by exerting gentle pressure at the posterior end of the hemipeneal bulge and rolling the finger or squeezing the fingers in an anterior direction. Here we can already see the base of both hemipenes being extruded. This will make the preparation much more likely to succeed. To fully extrude the hemipenes, we will first use a syringe filled with water. You can also do this with high concentration ethanol straight away, but it is less forgiving of mistakes. The syringe is inserted toward the back of the hemipeneal sac, oriented anteriorly so that the fluid is pushed into the hemipenis. It is useful to apply pressure at the hip joint to ensure that the fluid does not simply fill the body cavity, but is really directed into the hemipenes. Using gentle, constant pressure, fluid is then injected. Notice how, although the syringe is inserted only on the left side, it is filling both hemipenes. This does not always happen. You can further inflate the hemipenes by making additional injections. Once satisfactorily everted, the external surface is sprayed with high concentration ethanol. This fixes the organ in place. Then ethanol is additionally added from the inside. Now you can examine the surface structures of the hemipenes. The rotulae are visible toward the asulcal edge of the distal tip of the hemipenis. Here, on the left hemipenis, we can see the asulcal surface. In many species, it is covered in calices, a network of ridges that have a honeycomb-like structure. Rotating, we can now make out the sulcal surface. The sulcus spermaticus is a groove that transports sperm into the female's reproductive tract, and it is this structure that we use to define the sulcal and asulcal surfaces of the hemipenis. The hemipenis of Trioceras hunuli has two pairs of rotulae on its distal surface, one asulcal, as we've seen before, and one sulcal. Between them is a broadened, flat surface that has a field of papillae, which are best seen under the microscope. Our final subject is a corn snake, Pantherophis guttatus. As mentioned before, snakes were once thought to be an early branch off the squamate tree, but are now known to be one highly successful group of legless lizards. At present, it is not clear which modern group they are most closely related to. All snakes share a series of synapomorphies. They lack eyelids and external ears, have long forked tongues, an extremely extended vertebral column and rib cage, have one lung substantially longer than the other, and lack limbs, although some snakes, chiefly the Boidian snakes, retain a pair of cloacal spurs, which are remnants of hind limbs. All snakes are carnivores, and almost all species do not pre-process their food but swallow it whole. This is achieved through a unique skull arrangement that includes highly mobile jawbones. Pantherophis guttatus is a colubrid snake native to southern North America. It is one of the most popular reptiles in captivity. It is non-venomous and lacks fangs, instead using constriction to subdue prey. All squamates exhibit ecdysis, that is, they shed their skin. Snakes, in particular, almost always shed their skin in a single piece, which comes off inside out. Because scale arrangements and scale counts are extremely important in snake identification, snakes can often be identified to species level based on a sloughed skin alone. In most snakes, the two lower jaws are not strongly connected anteriorly, giving a great deal of mobility. In the mouth of the snake, we can see the large opening of the trachea. In the roof of the mouth, we can see that Pantherophis guttatus has two rows of teeth. The outer row is on the maxilla, the inner row on the palatines. In anterior view, a small notch is visible in the rostral scale of the snake. It is through this notch that the snakes pass their tongues for olfaction. Most snakes have a single broad row of ventral scales. In contrast, the underside of the tail has either divided or undivided scales. The cloaca can be identified by a larger flap-like scale. In some species, like Pantherophis guttatus, it is divided. This marks the end of the body and the start of the tail. To prepare the snake, we pin it in place. As it is long and lacks limbs, pins must be placed through the body. We take care to not puncture any vital organs by placing the pins to the sides. To begin the dissection, we make a transverse incision just anterior to the cloaca, and then run a sagittal incision up to the neck. We can then pin the rest of the body in place with the flaps of skin. This is a laborious process, but it is worth taking your time. Once the animal is fully pinned, the organs are clearly visible. The heart is located roughly 1/3 down the body. The liver is long and thin and runs beside the stomach, which is likewise long. A set of fat bodies occupy much of the posterior body. Pantherophis guttatus has a splenopancreas, located roughly 2/3 down the body. The kidney is near the cloaca. Snake lungs are asymmetrical. One lung is large and elongated, while the other is reduced. In some species, the reduced lung retains its function, but in others, it is lost altogether. To see the organs better, we can open the peritoneal membrane. We can then cut the mesenteries that connect many of the internal organs. The trachea is clearly visible as a ringed tube from the throat to the lungs. The hepatic artery is large and connects the liver to the heart. The kidneys again lie dorsal within the body cavity along the ventral surface of the spine. Once again, they are elongated. The stomach opens to the small intestine, which is tightly looped. The ureter and sperm duct run from the kidneys to the rectum. Snakes secrete urea primarily as a white, powdery solid. The testes are small and elongated, lying near the kidneys. With the internal organs removed, we can examine the vertebral and costal structure of the snake. Note that the vertebrae are largely undifferentiated. Each has a rib attached to it.
It's a long way from up the Yorkshires. It's a long way to us. It's a long way from up the Yorkshires to the meanest human cars. Well, it's goodbye to fins and gill slits, and welcome lungs and hair. It's a long, long way from up the Yorkshires